Chemotherapeutic agents are chemicals used to treat diseases. Among them are antimicrobials, which include antibiotics, biologically produced agents, synthetics, chemically modified antibiotics, and synthetic agents. Successful chemotherapy against microbes is based on selective toxicity, that is, using antimicrobial agents that are more toxic to pathogens than to the patient. Antimicrobial drugs affect pathogens by inhibiting cell wall synthesis, inhibiting the translation of proteins, disrupting cytoplasmic membranes, inhibiting general metabolic pathways, inhibiting the replication of DNA, blocking the attachment of viruses to their host, or by blocking a pathogen's recognition of its host. Beta-lactams, penicillins, cephalosporins, and monobactams have a functional lactam ring. They prevent bacteria from cross-linking NAM subunits of peptidoglycan in the bacterial cell wall during growth. Vancomycin and cycloserine also disrupt cell wall formation in many gram-positive bacteria. Bacitracin blocks NAG and NAM transport from the cytoplasm. Isoniazid, or INH, and ethambutol block mycolic acid synthesis in the walls of mycobacteria. Echinocandins block synthesis of fungal cell walls. Antimicrobial agents that inhibit protein synthesis include aminoglycosides and tetracyclines, which inhibit functions of the 30S ribosomal subunit, and chloramphenicol, lincosamides, streptogramines, and macrolides, which inhibit 50S subunits. Mupirocin stops polypeptide synthesis by binding to tRNA molecules that carry isoleucine. Oxazolidinone blocks initiation of translation. Antisense nucleic acid molecules also inhibit protein synthesis. Polyenes, azoles, and allylamines disrupt the cytoplasmic membranes of fungi. Polymyxin acts against the membranes of gram-negative bacteria. Sulfonamides are structural analogs, or para-aminobenzoic acid, PABA, a chemical needed by some microorganisms, but not by humans. The substitution of sulfonamides in the metabolic pathway leading to nucleic acid synthesis kills those organisms. Trimethoprim also blocks this pathway. Drugs that inhibit nucleic acid replication in pathogens include actinomycin, nucleotide analogs, fluoroquinolones, and rifampin. Chemotherapeutic agents have a spectrum of action and may be classified as either narrow-spectrum drugs or broad-spectrum drugs, depending on how many kinds of pathogens they affect. Diffusion susceptibility tests, such as the Kirby-Bauer test, reveal which drug is most effective against a particular pathogen. In general, the larger the zone of inhibition around a drug-soaked disc on a Petri plate, the more effective the drug. The minimum inhibitory concentration, or MIC, usually determined by either a broth dilution test or an E-test, is the smallest amount of a drug that will inhibit a pathogen. A minimum bacterial concentration, MBC test, ascertains whether a drug is bacteriostatic and the lowest concentration of a drug that is bactericidal. In choosing antimicrobials, physicians must consider how a drug is best administered, orally, intramuscularly, or intravenously, and possible side effects including toxicity and allergic responses. Some members of a pathogenic population may develop resistance to a drug due to extra DNA pieces called R plasmids or to the mutation of genes. Microorganisms may resist a drug by producing enzymes such as B-lactamase that deactivate the drug by inducing changes in the cell membrane that prevent entry of the drug, by altering the drug's target to prevent its binding, by altering the cell's metabolic pathways, by pumping the drug out of the cell, or by protecting the drug's target by binding another molecule to it. Cross-resistance occurs when resistance to one chemotherapeutic agent confers resistance to similar drugs. Multiple drug-resistant pathogens are resistant to 3 or more types of antimicrobial drugs. Synergism describes the interplay between drugs that results in efficacy that exceeds the efficacy of either drug alone. Some drug combinations are antagonistic.